Hello, everyone. So today we have David McKellar. He is a research scientist in the Technology Innovation Lab at the New York Genome Center. He received his PhD from the Department of Biomedical Engineering at Cornell University and his Bachelor of Science in Biomedical Engineering at Georgia Tech, and trained as a postbaccalaureate fellow at the National Human Genome Research Institute. David's research focuses on developing both molecular and computational tools to study the entire transcriptome, not just protein coding mRNAs. Spatial transcriptomics reveals the histological context of gene expression, but most methods only capture poly A-tailed transcripts. This limits detection to a narrow slice of the transcriptome, and to these ends, he has developed spatial total RNA-Sequencing, or STRS, and by adding synthetic poly-A tails to RNAs in situ, STRS enables capture of non-A tails RNAs with existing spatial platforms. And with that, I hand it over to David.
Thank you very much for that introduction. All right, let's share the screen and get going. Okay, so as Hector said, I work on building spatial transcriptomics technologies to capture transcripts which are generally overlooked by other methods. And so right now I'm working up in Manhattan at the New York Genome Center. Today I'm going to mostly be talking about some of the work I did while I was at Cornell working in Ben Cosgrove and Iwijn Vlaminck's labs. So let's jump into it. All right, so to start off, I think to kind of substantiate this work, right now, if you go through the latest release of human gene annotations from Ensemble and just count up how many genes are associated with different types of RNAs, protein coding genes are actually the most abundant, but every other type of RNA is actually non-coding, and they actually account for way more of the genes across the genome than protein coding. And so even though most of the technologies that people are using to profile cells and tissues only focus on messenger RNAs, there's a ton of other information there which should be used to understand gene regulation, disease development, all these other things in biology. And so spatial transcriptomics, just to kind of introduce, I don't think this audience necessarily needs this, but kind of the workhorse of the field over the past few years has been this technology, which has been commercialized by 10x Genomics, originally developed by the Lüneburg lab, called Visium. And so this technology uses these arrays of about 5,000 barcoded spots, and they're kind of spread across in this orange crate packing distribution on a glass slide. Each of those spots contains hundreds of thousands, millions of these single-stranded DNA oligonucleotides, which have spatial barcodes shown here at the bottom that can be used to spatially map the molecules back to the XY coordinates of the spot on the slide, and also include other features like these unique molecular identifiers, which can be used to collapse the sequencing libraries back down and make sure that you don't count the same molecule twice. And then the last feature on these oligos is a poly-T capture probe. And so this kind of gray bit at the end is used to capture and reverse transcribe RNAs in situ. The big problem here is that these poly-T capture probes were designed to capture just RNAs that have A tails. So that mostly includes messenger RNAs, which code for proteins, and a handful of long non-coding RNAs. But most of the transcriptome doesn't have poly-A tails. And so these technologies, which come in all shapes and sizes at this point, are completely missing most of the transcripts that are found in cells. So we set off to figure out a way to adapt spatial technologies to be able to capture non-coding RNAs and any other type of RNA that doesn't necessarily have an A tail. So to start, kind of building off of some other technologies that were developed at Cornell in the Vlaminck lab, I want to introduce this method called DART-seq because it played kind of a pivotal role in the development of STRS. So DART-seq took a slightly older single-cell RNA sequencing-based technology called drop-seq, which uses these polystyrene, really small polystyrene beads that have very similar barcoded oligonucleotides on them. And Redo and Phil came up with a way to append on these extra sequences onto the end so you could use a small fraction of the oligos from the bead to actually do targeted capture capture of specific RNAs. And so they use this technology to capture B-cell receptor transcripts and also viral RNAs. And that way they could enrich the libraries for these specific targets. And just to show you one of their results, so they took these DART-seq beads and designed 2 different constructs, one of which, shown in red, targeted all 10 of the gene segments from this virus. This is the Rio virus, which is an RNA virus. It's a kind of enough detail for that for now. But so in the red construct, they designed 1 probe for each of the gene segments from Rio virus. And for the blue construct, they designed a bunch of segments for just for one of the gene segments. And so if we look at like an IGV browser view of just the total reads across all of these different gene segments, at the top in yellow is the standard DROP-seq, which only has the poly-T capture probes. In the middle is the red construct, which targets each of the gene segments, and the blue is the targeted construct, which just goes for this S2 gene segment. And so they were able to show that using this targeted strategy, they could capture non-A-tailed transcripts with much higher sensitivity than the standard technology. But there was one really interesting finding in this initial effort with DROP-seq that kind of surprised me, where in some of these transcripts, which again, they don't have poly-A tails, but they were able to capture these kind of peaks somewhere in the middle of the genes. And this turned out to be because of short A stretches within the RNAs. And so we had this idea of instead of trying to rebuild the technology, we could start by going through existing single-cell data sets and kind of depend on this spurious capture of transcripts by short A repeats in them. So that would look something like this, where you have a short segment of A's within the RNA, which gets primed by the DROP-seq beads and then reverse transcribed. And so again, we had this idea to kind of mine existing single-cell RNA sequencing data. So most of this work was done during the pandemic. And so I essentially spent a large amount of time scrolling through the NCBI databases and finding all of the different single-cell RNA sequencing data sets that people had published. I specifically was interested in studying skeletal muscle regeneration. So all of these data sets were from mouse skeletal muscle. And we ended up with a fairly large data set that I spent a lot of time annotating and kind of integrating together into one place. And one of the interesting findings from this study was that there were a ton of reads from these data that were just completely ignored by existing analyses. And so to highlight another bit of work from the Vlaminck lab, work done by Michael Wang, to try to actually pull information from these transcripts, which were not which were completely ignored by existing computational tools. Michael developed this workflow called TAR-scRNA-seq, where TAR stands for transcriptionally active regions. And so the idea here was that when you take these RNA reads, you align them to the reference genome. A lot of them fall inside of genes, but many of them align outside of known genes. And so Michael developed some clever strategies to annotate these reads in a de novo fashion, so that these peaks that are outside of known genes, he could kind of, again, in a de novo fashion, build his own gene annotations. And then go through a very similar downstream computational analysis where you build these tables of how many copies of each gene are found in each cell. And he could split them across annotated transcriptionally active regions and unannotated transcriptionally active regions, and then use these count matrices to do things like dimensional reduction analysis and clustering analyses and look at all the different kinds of information. And so these plots here are showing two-dimensional embeddings, UMAP embeddings from a bunch of different data sets using different strategies of counting the RNAs, where the top is just looking at annotated genes. The second row is looking at unannotated transcriptionally active regions. And so Michael found that in some of these tissues, you can get really similar clustering patterns and dimensional reduction, UMAP embeddings from only looking at reads that fell outside of known annotations. And so I wanted to take this idea back to our single cell data set and see if we could do the same thing. And so going back to the 300,000 cells that I had pulled off of NIH's databases, here I'm showing one dot for each of those data sets. So each dot is something like 10,000 cells. And the x-axis here is showing what fraction of the reads from that sample align to the genome. And then the y-axis is showing what fraction of those reads align outside of known genes. So we essentially found that a lot of these data sets had 5% or so of their reads that were just getting completely ignored. They were aligning confidently to the genome, so there was, putatively some information there of RNAs that were transcribed and doing something in the cell. But again, the data just got completely thrown out. And so at this point, we ended up coming back into the lab, and I spent a lot of time thinking about how to use this data. It turns out that relying on short A repeats inside of RNAs is not a very robust way to discover new non-coding transcripts. And so this idea turned out not to work very well. But through the process, learned a lot about how how these analyses work and what types of information are actually inside of these RNA-seq data sets. So kind of reapproached the problem, and we asked another question, which is, instead of just trying to gather as much data as possible and mine that data for spuriously captured RNAs, how can instead we adjust the workflow so that we can enrich for transcripts which don't necessarily have poly A tails? So the idea we came up with was pretty simple. If they don't have A tails, then we should just add the A tail ourselves. So we came up with this method called Spatial Total RNA-Sequencing, which initially we built off of the Visium platform. And it's pretty simple. We just add one extra step into the workflow, where we start off with a fresh frozen sample, section it to 10 microns onto the slide, fix it with non-cross-linking methanol fixation, H&E stain it, image it. And then before we start the actual RNA-seq piece of the protocol, we add this one step where we perform in situ polyadenylation, where we add this enzyme yeast poly A polymerase, or YPAP, into the tissue. And this enzyme adds A tails to all of the RNAs in situ. So our messenger RNAs and some long non-coding RNAs, which already have A tails, those A tails just get extended with the synthetic A's that are shown here in red. Other RNAs like microRNAs or SNO RNAs, tRNAs, or even RNAs that are not from the host, like those viral RNAs that I showed from the DART-seq paper, those transcripts get polyadenylated, so they now can get captured by the standard Visium arrays. And the nice thing about this is that you really don't have to change a whole lot of the downstream molecular side of things. You can use just the standard PCR and fragmentation and sequencing strategy. The computational side of things is a little bit more complicated, as I'll kind of get into at the end, but generally the workflow is about the same. You only have to buy one extra enzyme, and you essentially turn on this light, which shows you the other half of the transcriptome. So what does this data actually look like? So the first experiment we did was taking some of these skeletal muscle samples that we were interested in. And these are mouse tibialis anterior muscles. And so the blue on top is showing a Visium experiment done with the standard workflow. And then the bottom in red is showing you our STRS strategy, where we do in situ polyadenylation. And so we're looking at this gene VALT-RC5, which is a non-coding RNA that is not very well studied. And no one really knows what it does, but I would argue that that's largely because people have not necessarily had the tools to be able to capture it. So if we look at the coverage across the body of the gene, this is, this arrow is pointing left to right. So it's on the forward transcript, which means it would be on top. And so in light gray here is showing coverage of the gene in the standard workflow. And so maybe we're capturing about a dozen transcripts in total. The y-axis here is log 10 transformed. But with our in situ polyadenylation strategy, we're able to capture, a few thousand transcripts instead of just a few dozen. And so we're, about 100xing the signal from this gene. And we can look at all sorts of other non-coding RNAs. So here we're actually looking at the RPS8 locus, which is a messenger RNA, but it's a really fascinating protein coding gene, which encodes one of the ribosomal proteins. And if you look at the intronic sequences, there's actually 4 of these small nucleolar or SNO RNAs, which are found and are encoded in different introns. And so in the standard assay, say if you look at SNORD55, which is here at the 5' end of the gene, as it's encoded on the antisense strand going from right to left, SNORD55, you can see in the standard assay is completely missing, but in our STRS assay, we get really great signal of about a few hundred transcripts. So this was really exciting. And the beautiful thing about spatial transcriptomics is that not only do we capture these RNAs, but we can also spatially map them to the location in the tissues where they're actually expressed. So if we take, these are the exact same data I was just showing you from the exact same experiment. Here we have the H&E images of the samples. These are 2 days post-injury, so you can see a bit of immune infiltration into the middle of these muscles. So this kind of area in the middle is the injury locus. If we look at GAPDH, the signal from both samples is almost identical. But if we look at some of these other non-coding RNAs, you can see that in the standard assay for spots that are shown as gray, the transcript was not detected at all. There were 0 molecules captured. But in our STRS assay, each of them is actually shown to be constitutively expressed across the body of the tissue sample. So again, we're just turning the light on for all of these genes and finding signal which was just completely missed in the past. So we can also look across the entire transcriptome and look at what fraction of the library is attributed to all of these different transcript biotypes, RNA biotypes. So protein coding is still the bulk of the signal across these data. But microRNA, again, goes from 0 to a few percent, maybe 2% of the total data. Long non-coding RNAs stay generally the same. That's because a lot of them actually do have poly A tails. One of the surprising results here was that we're actually able to capture tRNAs. This is surprising because tRNAs actually have an amino acid covalently bound to the 3' end, which is where the A tail would be added. And so we're probably capturing tRNAs that don't have an amino acyl group, but still we're able to turn the signal from very, very low to relatively abundant. And there are other RNA biotypes where this is a little bit the signal is amplified a little bit more, and a lot of these are small RNAs, small nucleolar RNAs, small nuclear RNAs, and things like that. So we were really excited to start to study all of these types of RNAs, which were again just completely missed by previous assays. So we went back to our skeletal muscle system and collected samples across this injury time course, where we start off with an uninjured sample, and then we inject the mice with a snake venom toxin called Natexin. And after 2, 5, or 7 days, we collect the samples and assay them with spatial total RNA sequencing. And so if we group all of these, the spots from the STRS spots together and cluster everything, then we can start to find the injury locus in the middle where you have immune infiltration, which is shown here in red, also in the day 5 sample. And then the border around the injury locus seems to look transcriptionally different. And so we're just calling this the injury border for now. And by day 7, the injury is almost closed, but you can still see remnants of where the injury was occurring in the sample. And so if we perform just simple differential gene expression across these 3 clusters, we can start to identify non-coding transcripts, which are specific, expressed in a spatiotemporally specific pattern across the tissue. And so let's say this first gene called MEG3 seems to be specifically turned on at the injury locus at day 5, which is an interesting time point because it's kind of an anti-inflammatory time point in muscle regeneration. And so that tells you a bit of some of the cell types that are probably expressing this gene. You can also look for other time points like the day 2 time point, which is much earlier and more pro-inflammatory environment. Or you can find transcripts which are expressed at both time points in the injury locus. And again, we're capturing many thousands of genes here. So there's a ton of analyses that you can do, and you can really keep digging through these data for months or years if you want to. So another one of the really exciting things is that we're actually able to capture mature microRNAs. And so this was surprising because these RNAs are only about 20 nucleotides long. I won't go into the details of the entire library preparation, but that's really surprising because even when you're just PCR amplifying these libraries, most of the transcripts which are that short are removed from the library just to kind of purify the library. But we were really excited to see strong signal. So you can look at mature microRNAs like miR1A, which is constitutively expressed across injury response in the muscle tissues, or miR206, which has been associated with fusing myocytes in late stages of muscle regeneration. And we found it to be specifically expressed in the injury locus at day 5. And another thing you can do is actually take single cell data sets. This is a really useful analysis for these Visium data sets where you can take a single cell data set, combine it with your spatial transcriptomics data set, and use the single cell resolution to kind of improve the quality of your spatial RNA sequencing data set. So here we used an algorithm called Bayes Prism, which is developed by Tinyi Chu, who has actually now joined the Vlaminck lab and is hopefully still working on these types of algorithms to improve spatial data. But with Bayes Prism, we're able to infer what fraction of the transcripts from each spot are associated with different cell types. So here we can infer the spatial distribution of fusing myocytes across injury response. And so you can see kind of by day 2, they really start to pop up at the edge of the injury. And by day 5, the injury is kind of starting to close in as you have this increased fraction of fusing myocytes. And you can find other cell types that are there and spatially co-localized with these fusing myocytes. Like here, these are fibrogenic adipogenic progenitors. So these are stem cell population, a bipotent stem cell population in skeletal muscle. And this specific subcluster we call pro-remodeling. These express a lot of proteases and other matrix-associated proteins. And so these cells are co-localizing to the injury locus and helping rebuild the tissue. One last technical kind of number here to show you that we are not diluting the library. So one worry we had when we were capturing all of these new transcripts was that we're potentially, only capturing non-coding RNAs like ribosomal RNAs, which are kind of constitutively expressed across all of these different cells. And we're potentially diluting out the signal from different cells. And so that turns out not to be true. So what I'm showing you here are these cell type fractions from Bayes Prism. Each dot here is a different cell type. Each plot is a different injury time point. So just showing you day 2, day 5, and day 7 after injury. The x-axis is the standard Visium assay, and the y-axis is our STRS data. And so we generally see that the cell type proportions across each of these data sets is almost identical. You can see the statistics shown here at the top. But this was exciting just to see that we're still able to recapitulate the same cell type specific gene expression repertoires, but also able to add in all of this information from non-coding RNAs. So most of my thesis work when I was at Cornell was done on skeletal muscle regeneration, but of course there were others around me working on other things. One of which was one of my close collaborators, Madhav Mantri, who was working on this fantastic project looking at viral myocarditis. And so Madhav was taking reovirus, this is the same virus that I showed you from that DART-seq paper early on, and working with John Parker's lab, infecting mice and then tracking the virus from the ileum all the way through to the heart and tracking myocarditis. And so Madhav used the standard Visium assay to look at what cell types are abundant at different time points across the infection process, and was able to show that about a week after infecting these mice, you get these myocarditic patches, which are shown here in orange. And then, like the injury, you get these regions around the patches where you have kind of similar changes in gene expression. So in the H&E, you can see the immune infiltration. There's a ton of T cells, NK cells, and things like that. By clustering these spots, Madhav was able to kind of infer where the virus was based on the host gene expression response. I was not satisfied by that. I wanted to actually see where the viral RNAs are, because again, reovirus is an RNA virus, and so if we can add A tails to the reovirus RNAs, then we can directly measure the viral RNAs. So Madhav gave me one of his samples, and I compared it to some of the data that he had generated. So this is 7 days after infecting the mice, and here we ran just the standard Visium or our spatial total RNA sequencing strategy. And what I'm showing you here is in gray undetected, and then in the color scale showing you the number of viral RNAs that are captured in each spot. And so this is a very similar pattern to some of the lowly expressed non-coding RNAs that we were finding, where by adding in STRS, you turn the signal from 0, shown here in gray, where there was not a single molecule collected, turn the signal from 0 to on, lowly abundant, but still on. So Madhav came up with this way of doing a targeted pull down of just the viral RNAs from the final library. And so he used these biotenylated probes to hybridize against the viral RNAs in the library and then pull them down with magnetic beads. And that turns out to be a really effective way to enrich for these viral RNAs and really deeply study the spatial distribution of the viral RNAs and the host response to viral infection. And so we can take the same data, cluster across the entire host gene expression response and get similar patterns like Madhav showed in his paper. Here I'm showing you an uninfected control in the top and then an infected sample 7 days post-infection here on the bottom row. Again, you can kind of perform differential gene expression across these two. And here we're able to find non-coding RNAs, which are associated with the host response to viral infection. So these are generally long non-coding RNAs that we found that seem to be really interesting, like AW11-2010. You know, all these genes with these crazy long names have crazy long names because no one has really studied them. And so these RECON genes, you know, are all kind of predicted genes from the RECON Institute. Some of the other interesting genes that we found in this differential gene expression analysis appear to be chemokines, but in mouse are actually not protein coding. So in humans, CXCL11 encodes a chemokine that's associated with, I think, T cells. I might be wrong there, but in mouse, it's actually not protein coding, and so we found a number of these chemokine-related genes, which turn out to be spatially co-localized with T cells. And so, just to confirm and make sure that this was a real transcript, we ran single molecule FISH, and so in yellow we're showing CXCL11 expression, and in pink we're showing TRBC2, which is a T cell associated gene. And so across this day 7 infected mouse heart, we can see spatial co-localization of these two genes, which kind of suggests that CXCL11 is coming from T cells. And then one last finding from this reovirus data that I think was really fantastic was reovirus is a double-stranded RNA virus. So its genome is encoded just in RNA, but in 2 strands. One of those strands is actually used as a genomic RNA, which is kind of a crazy term for me to come to terms with. But one of the cool things about this assay is we're actually able to capture both of those strands. So in blue, again, I'm showing you the Visium data. And in red, I'm showing you the STRS data. This is showing the normalized gene body position across all 10 of the gene segments from reovirus. So 0 is the 5' end, and 100 is the 3' end. So in red, we can see with STRS that we're getting really strong 3' capture of the forward strand, the positive strand. So this is the protein coding strand. And then the antisense gene, so 0 here would be the 3' end of the gene, and 100 would be the 5' end of the gene. And again, we're getting really nice 3' enrichment. And you see a bit of signal in the blue. These are those spuriously captured A segments from within the body of the gene. And then this peak right here happens to be, I think, 2 different reads that popped out of the Visium data, which just happened to be there. Probably noise from the experiment, but yeah, so this is, again, very exciting. So the last bit from this paper that I wanted to mention was we next had the question of whether or not this in situ polyadenylation strategy could work with single cell analysis. So all the data I've shown you up to this point was generated on the Visium platform, which only has about 50 micron resolution. So each of those spots contains something like 5 or 10 cells. And to really disentangle gene expression differences across these different cell types, 50 microns is not high enough resolution. So building off work from Steve Quake's lab, by Alina Isakova, and then also Alex Van Oudenarden's lab, we sought to see, if we could capture non-coding RNAs in single cells. And so I actually wanted to do single nucleus total RNA sequencing. So this data is just from a cell line, C2C12, which is a mouse myoblast cell line. So I was really interested in seeing if we could find regulatory RNAs from within the genome. If you remember back to some of that single cell data I had shown you before, single nucleus data is actually highly enriched with transcripts that align outside of known genes. And so I wanted to enrich even further. And so here I'm showing you expression of endogenously polyadenylated genes. In blue, again, is the standard assay. And then in red is our in situ polyadenylation single cell RNA sequencing or single nucleus total RNA sequencing. But again, this other half of the transcriptome just opens up. And so signal goes from basically 0 to highly abundant. And so here we're able to show, some of the first single nucleus total RNA sequencing data, which again, very exciting. And with these data, we can kind of build on some other analyses and look at, like I showed you with those viral RNAs, we can look at the normalized gene position. So because we're in the nucleus, a lot of the RNAs there are nascent RNAs and are being actively transcribed. So we actually see a really high enrichment for nascent transcripts in these single nucleus total RNA data, which is shown in this as this big 5' peak. And then we can apply Michael's UTAR analysis to find unannotated transcriptionally active regions across the genome, and see where, what other types of transcripts are being found in these nuclei. So in the top is showing you from genomic alignment where the reads align, and at the bottom is showing you the fraction of unique molecules which are associated with these unannotated transcriptionally active regions. So we see a huge increase in these UTARs as shown here in this bottom phylum plot. All right, so now that kind of is all of the data from the paper that I'm going to show you. But once we put this out, we next wanted to combine these two things. So not only do we want to achieve single cell resolution, but we also want to have spatial information in these data. So the next question was, how can we kind of achieve this? So looking at the Visium platform, I kind of already told you this, but these 50 micron spots really don't give you single cell resolution. So we next tried to find a different type of array that could actually get us to high enough resolution to find single cell gene expression values for non-coding RNAs. So we started with this platform from Curio Biosciences. This method is called SlideSeq. And so instead of these 50 micron spots, SlideSeq uses these 10 micron beads. So very similar to the DropSeq beads that I showed you earlier, but actually a bit smaller. And so they spread these beads out across a glass surface and then index each spatial location of the beads based on their barcodes, and then collect all of the beads and sequence libraries based on it. So you can get down to basically single cell resolution. The arrays are no longer perfectly distributed in this kind of regular pattern, but they're actually more densely packed in a random array. 10 microns technically in some places gets you single cell resolution, but if your cells are smaller than 10 microns or right about 10 microns, then there's a pretty decent chance that you'll have a cell that's overlapping two of these beads. So as we were starting off these new experiments, we were looking at all sorts of other platforms. So we also started to look at the StereoSeq platform from BGI, or I think they're now Complete Genomics. And so they call their platform Stomix, originally published as StereoSeq. This is an interesting one where you actually get much, much higher resolution. And so in the same area where you would have about 100 of these SlideSeq beads and fewer than 3 of the spots from the Visium platform, you get close to 40,000 of the pixels from the StereoSeq platform. Now, each of these pixels captures way fewer RNAs, so you generally capture about the same amount of RNAs in total. But here you can segment them out and actually attribute them to single cells. And so I'll show you some examples of that here in a minute. But if you zoom into a 10 by 10 micron square, which is about what a cell would look like, again, you can see Visium is, does not make the cut for single cell resolution. SlideSeq gets you pretty close, and StereoSeq gets you above that single cell level. So we decided to actually put all of these assays head to head and generate data on each of them and some of the same samples. And so this work I started while I was in the Vlaminck lab, but is now being led by Lena and Giannis in the Vlaminck lab. And so we adapted our assay, which initially we did on Visium, to these 2 new platforms. That we thought it was going to work right out of the box. That turned out to not be true, which I'll show you in a second. But eventually we were able to get it to the point where we can capture these data. This is from our virally infected heart samples. And here I'm showing you this gene miR6236, which is a microRNA, potentially has something to do with ribosomal RNAs. It's not very well studied, but again, the signal is essentially 0 in the standard assay, but quite high in these STRS assays. So I mentioned we had some troubles adapting this to SlideSeq. One of the first issues we had was that these beads, which are on the glass arrays, really do not like the standard fixation protocol. So we need to fix these samples so that we can perform in situ polyadenylation without the RNAs just diffusing across the tissue. we found that methanol fixation actually led to shearing of the beads. And so you get this really ugly, array. This is actually before you even capture the RNAs. But we were able to kind of tweak the protocol and change it to the point where we can keep all of the beads and actually generate high quality libraries with SlideSeq. So here's what the data looks like if you look at viral transcripts. So on the left, I have our SlideSeq data, and here I'm showing you those same plots from very similar plots from earlier, where the color scale is the fraction of RNAs that align to the viral genome for reovirus. So again, with our with standard SlideSeq, you essentially have zero capture of reovirus RNAs. And we were really excited to see that SlideSeq with in situ polyadenylation gives you actually quite abundant capture, much better than Visium actually, of these viral RNAs. Not 100% sure why yet, but still very excited to see these little myocarditic patches just by looking at viral transcripts. StereoSeq again goes from essentially zero signal to signal turning on, but it's not quite as abundant. We can also look at host non-coding RNAs, and again, we see similar patterns where in the standard assay, protein coding transcripts in the standard assay are something like 98% of the unique molecules, but with STRS, that number drops down to about 60 or 70% because the rest of the reads are going to non-coding RNAs. And we get kind of the inverse thing happening here with microRNAs, where the signal is very low and again goes to something like 1% in the final assay. And here the StereoSeq data, which I should have mentioned, we had to bin down to 20 microns, even though the actual array can achieve less, like sub-micron resolution. The signal from each individual pixel is so sparse that we have to bin them into 20 micron squares. But in those 20 micron squares, we can again see this reduction in the fraction of the library going to protein coding transcripts and this huge increase in microRNAs. And here in the StereoSeq data, we actually get really great microRNA capture, better than that of SlideSeq. And you can actually start to see microRNA myocarditic specific expression of microRNAs, which is kind of interesting. So we also wanted to test and see if this assay works on other tissue samples. So far, we had just worked on skeletal muscle and heart, which are both, kind of mechanically quite similar tissues. And so we've started this collaboration with Paula Cohen's lab. She's an expert in mouse testis development and all sorts of other things associated with male reproductive systems. And so we took these testes samples and applied first the SlideSeq. So at the top here, we have standard assay, and at the bottom, SlideSeq with our in situ polyadenylation. And we see similar levels of microRNAs in these data, which is really exciting. And then we repeated this experiment with StereoSeq, and here we're really able to achieve single cell resolution by performing segmentation on the data. This is actually led by Yannis Tekas, who applied cell pose to the DAPI images in the protocol, and then took those segmentations and added them onto the gene expression matrix so that you can bin all of the transcripts down to single cells. So each dot here is actually representing a single cell, and they're colored by different cell types. And so if you zoom in, you can start to see these different spatial structures. I've left the cell type labels out just so because there are kind of too many of them. It's a little bit too much information for one slide, but we're really excited to keep digging into these data because we can really achieve spatially resolved single cell total RNA sequencing. And again, we get really high capture of these microRNAs with the StereoSeq platform. Really high capture is a few percent, but because of the resolution, we're actually able to find cell type specific microRNA expression across these tissues. And so here are the cell types on the left, and this heat map is showing you a bunch of different types of non-coding RNAs, mostly microRNAs, and showing you the average expression for that cell type here from blue to yellow. And so really excited to keep digging into this data and find spatial, new spatial patterns of testicular development. So as I close, I just want to leave you with a couple maybe action items for the field. There are not a ton of us working on total RNA sequencing these days, but it is a growing field. So I'm really excited that other labs including that of Rong Fan, are starting to work on these total RNA sequencing methods. So Fan Lab recently put out this preprint for patho-dbit-seq, where they applied our in situ polyadenylation strategy to FFPE samples, so they can actually take clinical samples and collect total RNA from them. And there are a couple of really interesting technical improvements here, one of which is that they actually get really, really strong small RNA recovery. That seems to be because the RNAs are already so fragmented that they actually don't have to fragment the RNA as you normally would with an RNA sequencing library. So they can go straight into sequencing, and they get really high small RNA capture. Another issue in the field is that because it's kind of so young, there haven't been any benchmarking studies done across any of these methods. So here I'm showing you 3 different single-cell total RNA sequencing data. So our data, which is, I guess, actually single nucleus, data from the Quake lab, which is from a method called SmartSeq Total, and then data from the Van Oudenaarde lab, which is from a method called VASA-seq or VASA-drop. And I'm showing you the fraction of RNAs that are attributed to different biotypes. And so here I used a tool called Callisto to align all of them. The field hasn't really come to a consensus on how to actually handle the computational side of these data. And so we've been building tools to try to achieve this. I really just want to highlight the huge disparities across these different data. They all look totally the same, totally different, even though we are all claiming total RNA sequencing. Our data have a ton of these ribosomal RNAs because these other methods are doing ribosomal RNA depletion. I would argue that there's a lot of really interesting information in these ribosomal RNAs, but some people don't necessarily want to spend their sequencing dollars on them, which is a totally valid option. But the point here is that benchmarking studies and really rigorous analysis of how to properly count these RNAs are needed. And so to these ends, there are a couple things that I've been working on, one of which is a way to actually just manage genomic references. So this is a computational pipeline that I've been building, which, for now we're calling RefSnake. So building with a tool called SnakeMake, we, in just, 2 lines of code, you can grab all of your reference genomes and build new references with each of these different aligners so that you can keep all of your references up to date. This is actually a big problem in the genomics field these days, where reference genomes are updated quite frequently, but a lot of labs will stick with what they have kind of saved on their servers. And even though the quality of the references are increasing and new genes are getting added to references every year, folks are still using references that are 5 or more years out of date. And so they're really not taking advantage of the assets that NIH funds are paying for. And so this is my attempt to hopefully just make it easier for other people to actually access the tools that are out there. Another is a data pre-processing pipeline that I've been building, which we're calling SlideSnake for now. This one has a fun logo, but the handling these data is actually really complicated because there are a lot of different different artifacts that can be found. Coming from template switch oligos and adding in all these different non-coding RNAs makes it really complicated to align these data, like I mentioned before. So I've been working on this pipeline as a way to actually rigorously compare all of these different things. So I've added really rigorous adapter trimming to remove like PCR adapter artifacts, ways of computationally filtering out ribosomal RNAs so that you can directly compare different assays where one maybe had molecular ribosomal RNA depletion and another did not. And then also comparing different computational tools to align these transcripts to the genome, and a lot of quality control here at the end too. And so with that, I'd like to thank all the folks who worked on this, mostly Cornell folks here on the left from Ben Cosgrove and Iwijn Vlaminck's labs. Now I'm actually at the Tech Innovation Lab, and I really didn't get to talk about some of the work that I've been doing here. But we will have some really great preprints out, I think, in the next few months. And I'm very excited to keep building both molecular and computational tools to bring total RNA-seq to the masses. So with that, I'll take any questions.
Thank you, David, for the fantastic presentation. So are there any questions from the participants? Please feel free to unmute yourself or post on the chat.
Yeah, actually, one question. It's a great technology, by the way. So, and then especially comparing with the usual techniques like 10x and so. My question is, how sensitive is good in terms of connecting the data to the phenotype? Like, for example, a categorical versus continuous, a yes or no, or how low is good is connected to the myocarditis. Have you done any viral titers to kind of see whether this low is, or which ones? Of course, the spatial contact is very important where it's located, but is it correlated positively, or some of them not correlated? What would you comment on
Yeah,
that?
that's a great question. I believe for the original DART-seq work, they did viral titers, but we definitely didn't do that for the STRS data. This is a huge problem across the spatial transcriptomics field where people are, I think, constantly confused by, or really just don't quite misunderstand that. If a transcript is not necessarily detected by these assays, that doesn't necessarily mean that it's not there. I mean, you know, we really showed that by adding these poly-A tails to non-coding RNAs. But the current state of the art, you know, highest sensitivity sequencing-based spatial transcriptomic assays are really only capturing a few % of the RNAs. And so, you know, if your transcript is abundant or is lowly abundant, say fewer than 10 or 20 transcripts per cell, there's actually a pretty low probability that it's getting sequenced by one of these assays. So for you know, genes like that, I would suggest more sensitive assays, like targeted assays, like FISH and other FISH type assays. But yeah, we technically didn't do that analysis in this study, but based on the spatial co-localization, we can see that the viral RNAs that we're capturing do seem to be associated with the myocarditic regions. But yeah.
Thanks.
Wonderful. Are there any other questions? Please,
Yeah,
Cynthia.
sure. So we always run into this problem when we work with FFPE, right? We have these fragments of RNA. Now, I'm sure when you're cutting a 10-micron sections, you will get fragments of RNA that are created by the sectioning process. So the question that comes up is, how many of these fragments are natural fragments or versus artificial fragments created by transecting through cells? How do you differentiate the natural fragments from artificial fragments? And how do you know that, because you're not doing long read sequencing, so you're doing short read sequencing. So how do you know that these fragments truly represent the microRNA or non-coding RNA that are intronic in location rather than something that is really true? So yeah, you're getting the gist of my question, so I'll stop.
Yeah, it's a fantastic question. So the spoiler is that we are doing long read RNA sequencing. I didn't get to show any of that data today because it's still a little young and not quite ready for the limelight. But to better answer your question with some of the data that we do have in hand, so microRNAs are actually some of the only transcripts that we can resolve the mature short fragments versus the long fragments. And so you can we use a tool called Merge, M-I-R-G-E, where you align to either a precursor or the mature transcript. And based on, you know, where it aligns, you can infer whether it was a mature, you know, actually processed mature microRNA or not. But for longer RNAs, especially things like long non-coding RNAs, where there are, you know, sometimes dozens of different isoforms, which are, you know, processed in different ways, you run into a lot of issues. For those SNO RNAs that I showed you, which were intronic, those are nice because we actually do capture the 5' ends of those transcripts, and you get, you know, full coverage across the entire gene body. They're relatively short. I mean, we're doing like 100 bases of sequencing for these data. But yeah, it gets kind of hairy when you start to look at say tRNAs, which are in the like 120 nucleotide range, or anything that's a little bit longer than that. Where you really don't capture the whole transcript. And there are even more artifacts there where small RNAs and long RNAs, because of the way these assays are performed, you have all these different types of biases. So When you run the initial PCR amplification, small RNAs are really highly preferred because they're shorter and you have length biases in PCR amplification. But later on, when you fragment the libraries, this is something nice from Rong Van's lab where they don't have to fragment the libraries. But if you have 1 kb, 2 kb long cDNA fragments, then you have to chop them up so that all of your libraries about the same size for Illumina sequencing. That process actually highly prefers long transcripts because if the short RNAs get fragmented, then you lose the PCR adapter, and so you can no longer PCR amplify them. So you have kind of these forces that are pushing against each other and causing, opposing biases, which again are some of these technical things that I came to the Technology Innovation Lab to really try to dig into and figure out. But yeah, excellent question.
Wonderful. And then we have a question on the chat. Kristen Asensio is asking, is it possible to co-localize RNA binding proteins by antibodies with small RNAs of interest?
Yeah, that's an awesome idea. I would point you to some of the new multi-omic strategies that folks are coming out with, like spatial site-seq or SPOTS. Again, another Rong Fan paper, or SPOTS from the Landau lab here at the Genome Center, where they're taking site-seq, where you have these antibodies that have barcoded oligos appended onto them, and you can bind your protein of interest with those antibodies and barcode them so that you can add the protein information into these libraries. I haven't had a chance to combine it yet with STRS, but would love to do so. If you want to do it, let me know. I'm happy to give you advice on how to run STRS, but there's definitely a way to do it.
Yeah. Are there any other questions? Okay, so I have one question, which is, you have a diverse RNA molecular, information, right? Could you also consider doing more dynamics like RNA velocity, implemented these kind of analytics, like from micro to intron to mature, and how they're spliced out? I think there's very rich data here.
Absolutely, yeah. And so one of the nice findings that didn't have time to highlight, but we actually do have a much higher capture of intronic reads in STRS than we do in the standard assays. I would point you to the VOSA-seq paper from Alex van Oudenaarden's lab, who did a much more rigorous analysis of this, and they actually find that in their single-cell total RNA-seq data, the RNA velocity analysis is much more sensitive when they add in this polyadenylation strategy. So there's a lot of cool biology to be done there. I'm really excited to do these types of analyses with long-read sequencing, where you have much better coverage of introns. I really don't trust short-read sequencing for splicing analyses, because for most genes you're missing most of the introns. But yeah, there's definitely a lot of runway.
Wonderful. Any other questions? Okay, wonderful. Then I think in the interest of time, let's thank Dr. McKellar again. I'm going to use this clapping sign. Where's my clapping sign? To congratulate. So I think here, reactions. And congratulations. I know this is virtual and it's hard to do. Congratulations, right? This was a great presentation. And also, before we wrap up, I'd like to also announce that in 2 weeks from now, June 14th, Dr. Alexander Shu from Cedars-Sinai will be presenting spatial environments for clinical biomarker development and therapy predictions. And again, thank you, David.
Thanks for having me.
Wonderful work. Thanks.
Wonderful. Then have a great one, everyone. Hope to see you in 2 weeks.